This video tutorial is all about chloroplasts and photosynthetic pigments. Hopefully you're already aware that chloroplasts are the organelles within plant cells where photosynthesis takes place. Algae have chloroplasts, but photosynthetic bacteria do not. Most plant chloroplasts are disc-shaped and around 2 to 10 micrometers long. Each is surrounded by a double membrane, the envelope, with an intermembrane space of width 10 to 20 nanometers between the inner and outer membrane. The outer membrane is highly permeable. There are two distinct regions visible on electron micrograms inside a chloroplast. The fluid-filled matrix called the stroma, as well as the grana that consists of stacks of thylakoid membranes. We're going to first of all focus on the grana. The first stage of photosynthesis, which is the light-dependent stage, takes place in the grana. Chloroplasts have three distinct membranes: the outer, the inner, and the thylakoid, giving three separate internal compartments: the intermembrane space, the stroma, and the thylakoid space. The thylakoids within a granum may be connected to thylakoids within another granum by intergranal lamella, also known as intergranal thylakoids. The thylakoid membrane of each chloroplast is less permeable and is folded into a flattened disc-like sac called thylakoids. These form stacks. Each stack of thylakoids is called a granum. One granum may contain up to 100 thylakoids. With many grana in each chloroplast, and with many chloroplasts in each photosynthetic cell, there is a huge space or a huge surface area for both the distribution of the photosystems that contain the photosynthetic pigments that trap sunlight energy, as well as the electron carriers and ATP synthase enzymes needed to convert that light energy into ATP. Proteins embedded in the thylakoid membranes hold the photosystems in place. The grana are surrounded by the stroma, so the products of the light-dependent stage can easily pass to the stroma to be used in the light-independent stage. The stroma is the fluid-filled matrix. It contains the enzymes needed to catalyze the reactions of the light-independent stage of photosynthesis, as well as starch grains, oil droplets, small ribosomes, similar to those found in prokaryotic cells, as well as DNA. The loop of DNA contains genes that code for some of the proteins needed for photosynthesis. These proteins are assembled at the chloroplast ribosomes. Within the thylakoid membranes of each chloroplast are funnel-shaped structures called photosystems. These photosystems contain photosynthetic pigments. Each pigment absorbs light of a particular wavelength and reflects other wavelengths of light. Each pigment appears, to our eyes and brain, the colour of the wavelength of light it is reflecting. The energy associated with the wavelengths of light captured is funneled down to the primary pigment reaction centre, consisting of a type of chlorophyll at the base of the photosystem. Chlorophylls are a mixture of pigments. All have a similar molecular structure, consisting of a porphyrin group, in which is a magnesium atom, and a long hydrocarbon chain. We're now going to look at chlorophyll A, and then we'll look at chlorophyll B. So there are two forms of chlorophyll A, both of which appear blue-green. Both are situated at the centre of the photosystems. Both absorb red light, but they have different absorption peaks. So P680 is found in photosystem 2, and its peak of absorption is light of wavelength 680 nanometers. In comparison, P700 is found in photosystem 1, and its peak of absorption of light is of wavelength 700 nanometers. Chlorophyll A also absorbs some blue light of wavelengths around 440 nanometers. Chlorophyll B absorbs light of wavelengths 400 to 500 nanometers and around 640 nanometers. It appears yellow-green. Carotenoids absorb blue light of wavelengths 400 to 500 nanometers. They reflect yellow and orange light. Finally, xanthophiles absorb green and blue light of wavelengths 375 to 550 nanometers. They reflect yellow light.